Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I wanted to let you know that we are working on a new program geared towards transitioning from academia to industry. Powered by Dr. GPCR, our new program is called the Life Career Job. The goal is to support you in your efforts to find your next job in industry, plan out your career trajectory, create a life, and support the lifestyle that works for you. If you'd like to be notified when this program becomes available, fill out our short survey on drgpcr.com/career. We are also launching our GPCR consulting services. Soon you'll be able to find the profiles of our carefully selected consultants on our website at drgpcr.com/consulting. You'll be able to find the help you need for your company. Stay tuned. The second edition of the Dr. GPCR Summit will be held between September 13th and 19th, 2021. We're planning a combination of live talks, pre-recorded talks, and live workshops. Visit drgpcr.com/summit for more information. Are you subscribed to our YouTube channel? If not, please subscribe today. It's not only a great way to catch up on our recorded events, such as the Dr. GPCR Virtual Cafe, but it's also a great way to support us here at Dr. GPCR. Hello, everyone. This is Yamina from Dr. GPCR. I am delighted today to have with me Dr. Stephen Foord. Hi, Stephen. How are you?
I'm fine, thank you. Hello, Yamina.
Thanks for being here. First, I should ask you, would you prefer Stephen or Steve? Which one do you prefer?
Steve.
It's the simplest. So I'm really excited to have you today with me. We're talking about GPCRs, and when I contacted you, you were mentioning that you don't really work in the field anymore. However, I looked you up and you had made important contributions to the field. So can you tell the audience, who are you and what do you do right now?
Who am I? I started off working in academia with a chap called Professor Reginald Hall, who sadly died before his time. He contracted primary amyloid, which was a tremendous shock for all of us. But I started in Newcastle, and it was a neuroendocrine lab. And I did mention to you, I was one of the first people in the world to get somatostatin and TRH injected into me. There were collaborations, Reg Hall had collaborations with the Shalley and Gilliman labs. That's what started off my interest in TRH and in neurohormones and GPCRs. At that time, of course, I was just worried, what were the side effects going to be? Was I going to die? Was I having palpitations, whatever? And I wasn't thinking, one day I'm going to be working on those receptors. So that's what got me into it. Reg Hall, as I mentioned, sadly contracted amyloid. The lab broke up and I got a Medical Research Council training fellowship to go work in molecular biology. And I elected neurohormones again, Roger Craig's lab at the Middlesex. And he was one of the discoverers of CGRP. And I joined his lab. He'd already discovered it. I sort of came in on the tail end of it. And he thought, wouldn't it be fun to work on the CGRP receptor? And I've been rather naive, thought, great, would that be fun? And the consequence of the fun was collecting placentas from Queen Charlotte's Maternity Hospital for about 4 or 5 months. If you've ever collecting a bucket full of placentas on the district line is an interesting experience, especially if you come across a guide dog.
Oh goodness!
Which has tremendous interest in the bucket. But we purified the receptor, and later I went on to clone it not by purifying it and doing protein sequencing, but I was well and truly hooked on GPCRs. But that lab broke up because of politics between Middlesex Hospital and University College. And I thought, I'll have a really stable job and go and work in industry. So I joined Glaxo, and then I joined Glaxo Wellcome, and then I joined GlaxoSmithKline, all in procession. So that's what most people would know me for. would be working for Glaxo in drug discovery, although a large chunk of what you do is it's not reported, it's not published. And so this is a sort of an interesting chance. So to put the record straight a little, for example, when I started work, I worked for nearly a year on HIV.
Wow.
And just about everybody worked on HIV. It was all hands to the pump, get a much needed drug quickly. So, we had a very large team. I worked on HIV TAT, which is a horribly difficult to fold multiple disulfide protein. And I ended up expressing it in xenopasuicides, which I have had an abiding love of ever since. Not HIV TAT, the oocytes. Yeah. And so we managed to get that to work. We knew how it worked. But again, we had drugs from other means. They didn't need a TAT-TAR inhibitor. So we moved on molecular pharmacology after that, because I had a degree in pharmacology and I had molecular biology. So that made me fairly unusual.
Interesting. You were mentioning that you were at GSK and all its different forms. Was this in the UK? Where was it? The reason I'm asking is because I was talking with Terry Kinakin exactly a week ago, and he also went through all the variations of GSK at this point.
Yeah, I know Terry Kinakin very well, and we were very lucky to work with him. What a nice chap.
He is.
Yes. I started in Greenford. Actually, no, I'll go right back. I started I was given a job offer working in Geneva at the Glaxo Institute of Molecular Biology by Jonathan Knowles. And I was very keen on working there. My wife at the time was not keen on going to Switzerland. So I took 75% pay cut and worked in Greenford in London on very much the same sort of job. So I started in Greenford, then I was moved to Ware, which was the big drug discovery. That was Pat Humphreys and sumatriptan and Mike Tyers and Dan Citron and pure pharmacology. So I was the outlier brought in as the molecular biologist. And then Stevenage. But I used to go to North Carolina quite frequently to see Terry, for which we got a sort of a preview on how to think. And it seeps in without you ever actually understanding cubic quaternary models. And you all of a sudden realize that you actually do know what's going on without you really realizing how you do. And then I spent a while in Philadelphia and Upper Merion working for Glaxo then. So I've been around a bit, but it's here I am now, but retired.
Still, you had quite an interesting set of accomplishments and quite an interesting set of discoveries. Let's talk a little bit about CGRP. You were mentioning the story about the placentas and getting the buckets of placentas. Where did the idea of collecting placentas and purifying the receptor from there come from?
That came because we did a series of binding experiments to see where the most CGRP receptor was. And it's not just where it was, but where was it that you could actually get literally a bucket full? It was in spinal cord, but you couldn't get a bucket full of spinal cord and so on. It was in placenta, but not quite as you think. Placentas have got, for want of a better word, interdigitating villi from the fetal and maternal sides. If you actually subject them to osmotic shock, the villi snap off. And you can throw away the rest and just keep these vascular villi.
Interesting.
And they were very high in CGRP receptors. So I could get quite a lot human receptor from a readily available source. Some people like to keep their placentas, but most don't. And so that's where we tested just where you could get it from. And we actually got the whole thing generally right. We got the molecular weight right. I got some purified. Sadly, when the amino acids sequence, we tried to determine it, the ICRF in London, that's the Imperial Cancer Research Fund, which Mike Waterfield was head of, at the time, it was struck by lightning just when my sequencing run was being done. So I lost 80% of my sequence and several months' work. But I did win the Agnes Ogden Memorial Prize for the best paper from University College that year. So it was all worth it.
It's interesting that it was, I never ever would have even thought to think about placentas when it comes to purifying a GPCR.
No, well, it's a vascular tissue. And yeah, that's, I guess, I've not seen any literature on it ever since.
Interesting.
But that's where we got it from in the first place.
Interesting. And then how did you go from purifying it to actually realizing that it's a combination of a ramp and a receptor?
No, we never did. We never made that jump. All we got was 66,000 molecular weight protein that looked like a dimer. Looked like there were two bands on the gel. Sometimes the higher band was more prevalent, sometimes the lower one. We had something at the bottom of the gel, which I always thought was some sort of impurity or rubbish, which was almost certainly going to be the ramp. So I just ignored it. I thought, what's this very consistent impurity I'm getting here? Very small. So we didn't get it. I just knew its molecular weight. And then I joined Glaxo, and for the next 5 years or so, I worked on everything but CGRP. And then the migraine program group came along and said they were having real trouble expressing CGRP receptors in the cell line. They hadn't got it cloned, but there's a cell line called SKNMC, which is a glioblastoma-derived cell. And they would grow the cells up in one of the cell culture cubes on a massive scale. And sometimes it worked, and sometimes they got no yield at all. And it was the screening groups they'd schedule a screen and have nothing to screen. So, they said, why don't you screen it? And that's the thinking, was you screen things because it was convenient. You cloned it, not because people had a desperate desire to know what the target was, because they thought their target was going to be completely conventional, GPCR, no big worries. But we didn't know anything about it then.
And how did you go from this stage where you didn't know anything about it to actually identifying what a protein complex it was?
Luck, as it usually is. You must hear so many folks on here say, oh, it was lucky. We knew it stimulated cyclic AMP, and we knew that from my oocytes, which I've already mentioned, which I did the HIV work, I knew that you could tie cyclic AMP through the cystic fibrosis transporter to a chloride channel and use electrophysiology. And the major thing in expression cloning is where you put a library encoding receptor, you try and pull out a pool that contains the signal you're looking for. The more sensitive it is, the more chance you've got of picking that one clone in 20,000 that uncovers itself. I went for sensitivity, and I went for SK and MC cells because the receptor was routinely there. It just seemed to disappear on scale up. So we did that. What we did realize was that there was already a CGRP receptor in Xenophus oocytes.
Wow.
But it was in the outer, they have a little layer around them that maintains it. The oocyte is actually one cell. It's got thousands of cells around it that maintain it. And you strip those thousands of cells off, and those cells are supposed to be, it's like peeling a grape.
Wow.
Slippery one. I've not peeled many grapes, but you get rid of the outer cell, and then you thought, I could get rid of the endogenous receptor, and then I'd just detect what I put in. What it turned out was what I'd put in was RAMP1, and it was so powerful, it was potentiating the receptor that was already there. So the lucky thing was that human RAMP potentiated Xenophus CRLR into being the CGRP receptor. And the signal was enormous. It's the sort of thing that when you've spent years and years working on, grubbing around with statistics, when you actually see something where the chart recorder goes wild and you keep having to turn the amplification down, you realize that you've got something quite serious.
That must have been an excited moment. How did you celebrate, but how did you realize that you were making a discovery, basically?
Well, once we chased it down, because we formed a little grid of which pools had it, it was exciting, especially when we saw what the gene was. It wasn't a GPCR, it was the one transmembrane protein. And half the lab were convinced it was the best discovery ever, and the other half thought it was a total artifact and it was going to be really embarrassing.
Interesting.
And I think one of the things you get from that is you need a combination of both in the lab. Yeah, sort of heady optimists and hardened cynics. And the effect was so massive, and it explained, we knew immediately what we'd got. As soon as you do bioinformatics, you realize there was a family of 3 ramps. We knew that CRLR looked like the calcitonin receptor. We still had the adrenomedullin and amylin receptors to identify, and there weren't any more calcitonin-like receptors, but with the ramps, we could explain all the subtleties, or at least most of them. So, and that was, the whole thing was done within 5 months, maybe 6 months at most.
Wow.
And we published every experiment we had, everything. I sent all the reagents out to, people who are my friends now, Patrick Sexton, Kathleen Carone, Jan Fischer and Walter Bourne, who are in Zurich, Debbie Hay. They all got the clones, and Michel Bouvier. And they carried on, but we had another project to do. So we just closed, Glaxo at that point had lost interest, or they were chasing a Merck compound rather than anything else. And it wasn't good news for drug discovery, because the protein that everybody thought that small molecules were going to bind in a GPCR binding pocket.
Yeah.
But if the binding pocket was just the same for amylin, adrenomedullin, CGRP, this wasn't the best of news. Yeah. Nowadays, we know those compounds could be binding pretty well anywhere.
Exactly.
Yeah, lots of targets all over the place, grooves, things that's stopping the receptor flipping from one conformation to another. But then, how are we going to get selectivity when the target's exactly the same in at least 3 receptor classes?
That's interesting. I think at that point, maybe leadership didn't understand that nature is trickier than that. And just because it's the same GPCR. Yeah.
Well, I think leadership thought, probably reasonably, that they had more chance of making an analog of the Merck CGRP antagonist than they had of figuring out selectivity, because theirs already was selective.
Yes, that was.
So they thought, they could. Now, without going into it, there were lots of problems with CGRP antagonist small molecules, liver toxicity being one. And then, of course, completely out of left field come antibodies directed at the CGRP complex and against CGRP itself. Yeah. And they have essentially become the frontline treatment for at least one flavor of migraine now.
Yes.
So, and I'm embarrassed to say that I did not immediately think, I should rush out and patent this. I could make a small fortune selling CGRP receptor antibodies, because there weren't many. There still aren't many GPCR antibodies. And the ones we have actually are autoimmune disease, the ones we don't want, not therapeutic. So we stopped, or we were told to stop the project, and did forthwith, like a good company person should.
And then you went on working on HIV? Or this was
No, HIV was long in the past. No, we were introducing molecular pharmacology into Glaxo, basically. So we started off working. One of the first ones was angiotensin project,
okay,
which was tricky because there was one human receptor and two receptors in rats, and the people just did not believe that the second human receptor wasn't there. It just had to be that we'd missed it somehow. And of course, you can't go into a discovery program. The two rat receptors were so similar. Why wasn't there a second human one? And where was it? And we were going to have selectivity problems and whatever. Yeah. So I was tasked with tracking down the second, now we know, non-existent angiotensin receptor, the AT1 receptor. And we did that by doing southern blots, some mystical things now, southern blots, DNA chopped up. We did southern blots, looked where the bands were that hybridized, and there were two other things that looked hybridized to the angiotensin receptor sequence. One of them turned out to be what was then an orphan chemokine receptor, and the other one turned out to be the prostaglandin EP4 receptor. There was no other angiotensin receptor, which we know now, but at the time, the pharmacologists were distrustful of molecular techniques because there's potential artifacts in cell lines. You have to go through the orphan receptor literature to see it's riddled with artifacts, things that amplify up things that you aren't expecting. If you go through the IUFAR receptor orphan page, you'll see there's 35, 40 papers cited where people haven't been able to repeat it. And I've been that person. When we were doing this angiotensin work, it was published that the mass oncogene was an angiotensin receptor.
Wow.
And now there still is evidence that angiotensin 1 to 7 will activate it, but it's not a very good responder. But in our hands, angiotensin would, in fact, in a highly amplified system, it would work, but just not when you got the real angiotensin receptor. It was a thing of wonder. It worked fantastically well. It made you realize you were grubbing around in the noise when you had the real one. But at that time, things were a little more fraught than they appear now, and most people have passed that.
I think it's also an evolution in the technology and in all these systems that allow you to measure receptor function, and cloning has become a routine thing, and computational technology also has evolved, computational biology, and running. I don't think there are a lot of people around the world who actually run southern blots or DNA gels anymore. I don't remember having done it maybe in class back in undergraduate school, but that's about it.
Yeah. No, I should have learned to thatch roofs and do medieval stained glass. I'd probably be more in demand. I could sequence 600 bases a day, wow. Which fantastic. Now I think that's probably a few seconds work on the average machine. But the thinking still, still relevant.
It is definitely. And I think when I hear stories like you're telling about CGRP and the angiotensin, I think as a newer generation, we forget that for a long time people didn't even know that what a receptor was. For a long time they thought it was just a concept, and it turned out to be a protein. It was. Well, I mean, go back into the 1930s. It's an active principle. It's those guys didn't know what it was, but they didn't get it wrong either. And I think that's the trick. Every generation thinks, they've got a really hard job. But then when you look back, the previous generation thought it was a really hard job, and the one before that. And so I wonder how we've done so badly at the orphan receptors. I mean, I haven't done any work on them other than
browsing databases for 10 years, and there's really not. There is progress. It's being whittled round the edges slowly. There are still some big families which have conserved right down to fish, and we still have no idea what the ligand might be.
Exactly.
And the ligand gives you the pharmacology, the physiology, the potential disease links, and it's all just waiting to be found. You touch on things, how much you can find out now. When I've gone around posters at Gordon conferences, which I occasionally go to, there's so much data on them. There's just masses of information that you can find, but still often not quite the key piece you need. You can do a phylogenetic analysis from aardvark to zebra just on the database, but you still can't find out what the ligand is, or even get close to it.
There's a lot of work to be done, and I think that's why the field is still so busy, and there's those movements. People come into the field, work on various receptors, and use the current technology to screen thousands and thousands of compounds a day, and there will still be a need to advance our knowledge. And I was just looking at a paper, I think it was published last month, about the potential of orphan receptors as a therapeutic drug target. But then again, the problem remains, if you're an orphan, you don't know what ligand binds that receptor, which, as you mentioned, it is true, it gives the physiology and the pharmacology of it.
There's such a difference as well. The difference between a lead and a tool compound is still pretty massive. And the difference between a tool compound and a drug is more massive still. And we have still had lots of discussion. Oh, we've got a compound, this could be a drug. And you think, well, actually, you've no idea how far you're away from that. The attrition rate is just enormous. Most people who work in drug discovery are just delighted if they just get a sniff of the drug, that they did some. I'm really, really pleased. It's the best thing I've ever done to be associated with CGRP and migraine. My other big one was which is not that big. It's the EP4 receptor we cloned when we were looking for the angiotensin subtype. That prostaglandin EP4 receptor, that is one that, again, we actually had a compound at it. It was really good, very effective, weak partial agonist for rheumatoid arthritis. Now it's used in animal health for treating dogs with rheumatoid arthritis.
Interesting.
As a dog lover and the owner of a dog who had rheumatoid arthritis, I was very pleased about that. But that's the closest, that's the closest you get. There's a 35-year career, and you're happy that Labradors have got an easier walk in the morning.
Yeah, I think after talking to a couple of people in industry, and everyone who was even remotely associated to a drug that ended up either in humans or in the clinic, at least, I hear that it's a great feeling to have contributed to that. And I can echo the love for dogs. And I do have a big dog who's also suffering from rheumatoid arthritis. And
yeah,
it's
it wasn't my compound. I think it was a variant of it for Eli Lilly, but the patent made the link, which was another very nice thing to do. I don't actually know why it didn't progress into man, because the drug was sold off as part of a package to another company.
Interesting.
So, and that's something else that happens in industry. Things, you discover something of worth, and the company can't do that, or it feels that rheumatoid arthritis is pretty well treated, which it is, although possibly not so in dogs.
It is a problem in humans, but also in dogs. I can attest to that. So what did that compound do, that EP4?
It was a weak partial agonist.
Interesting.
It was interesting because it was discovered. We had a tremendous prostaglandin expert called Bob Coleman, who knew everything there was to know about prostaglandin receptors, GPCRs. And he had a compound that worked in pain models, but he thought it was an NSAID because it didn't work in any of his receptor models. He couldn't work out. How come it worked like a prostaglandin receptor blocker, but didn't work on any of the models he had? And when we cloned the EP4 receptor, I tested his compound. So his suspected NSAID wasn't an NSAID at all. It wasn't blocking prostaglandin synthesis. It was actually blocking the prostaglandin action on the receptor. So we tied in his activity with our molecular target. And of course, we knew it should work. And lo and behold, it did. But as you know, I think everybody knows, prostaglandins are associated with maintenance of stomach lining, helps the gastric erosion with aspirin. So that was always the worry because EP4 is in the stomach lining, and that was a concern. And now there's actually quite a significant literature of EP4 on cancer, colon cancer in particular. So it might show up again outside the world of veterinary health.
Who knows? And what led you to test that compound and specifically? Was it through discussions or?
Particularly because Bob Coleman had shown it worked. It worked in his animal models of pain, and we had really good support from the neurosciences division in Glaxo to test it in their models of pain, their models of rheumatoid arthritis. People were very generous with it. It was at the time competing with Vioxx and Celebrex, the COX-2 inhibitors. So, there were a lot of folks thought, as we know, that didn't necessarily end well. Although, in fact, we now know that ibuprofen has reasonable COX-2 activity as well. But at the time, they fell outside what was the risk profile of what an NSAID should be. So there's always some degree of politics in this, even within a company. What model are you going to split someone's market? Are you going to lose focus? Basically, it's very, very easy to lose focus, and warning off going chasing prostaglandins when you want a synthesis inhibitor. That's a good warning.
It is. And I think at the end of the day in companies, yes, it's important to make the discoveries. It's important to get that drug, but still it's a business and you need to make smart decisions in order to push forward any potential drug that will, at the end of the day, not only help people, but also bring in revenue to continue going through that cycle.
Yeah. Half an eye on how you're getting paid.
Yes, always. So I was looking at the bio that you shared with me, where we've talked a little bit about EP4. Where do GABA B receptors come into play? So what happens after the EP4 project kind of ends? We had a little golden 8-9 months where we'd identified a whole string of orphan receptors, mostly from bioinformatics. So I would identify them by bioinformatics, thought they were interesting, and sometimes we were given specific tasks. So GABA B came about because we had a yeast-2 hybrid expert, Julia White, and we had to come up with a good project for one would fit her skills, and her skills weren't being used correctly at the time. And so Fiona Marshall came up with the idea of, the reason GABA A doesn't work, the GABA B receptor doesn't work very well, is it's missing an accessory protein. Yep. And so it was, but the accessory protein was actually another 70M. So that was another running down the corridor, because we already had that other 70M ready cloned in the freezer because we thought it was really interesting. So that was a nice story. I think Fiona's already mentioned the carboxylic acid one, where
that turned out to be the vehicle that the test peptides were dissolved in rather than the actual peptides. So we found that everything dissolved in acetic acid was positive, irrespective of what it was.
Yeah.
That was Andrew Brown identified that. Nicotinic acid, when we were told there was a really interesting receptor for nicotine in both spleen and liver, I think. I can't remember exactly. It was in 2 particular tissues. Out of all the orphan receptors, there were only 10 that were in high levels in those 2, 20 even if you stretched it. So Alan Weiss tested all of them, and one of them was it.
Interesting.
So it's low-hanging fruit in a way, but we just seem to make the correct, lucky decisions 4 times in a row. I should have bought lottery tickets.
And patented the ramp.
Yeah.
The ramp.
Yes, I've done that as well.
But then so you'd mentioned that you went on from project to project, and each time you went on either screening or you had a problem, you had to come up with a new strategy to better understand a receptor. But what was the rationale behind it? Working at a private company, there must have been a link to some disease, or where did that direction come from?
Yeah, some of it came. So CGRP came because the migraine group. There was a substantial literature on that. Carboxylic acids came from nowhere. That was just pure serendipity. Free fatty acid receptors weren't even thought of at the time. Nobody asked for that. Nicotinic acid or niacin was a request. The cardiovascular the chap called Nick Pike, who was from, it said, we would really like this as a potential therapeutic. So we identified that to order. And GABA B was Fiona's idea because there was a significant literature that there was more than one GABA B receptor, and baclofen wasn't a particularly good drug. And if we could find more, then we could make a better drug. And as it turned out, there were no more GABA B receptors. We almost killed that literature stone dead because we couldn't find any more. So that one was closed. So that's where they came from. 2 requests, 2 serendipities found, and I say probably 3 when we got the EP4 prostaglandin receptor. That was a 3rd serendipity that got closest.
So basically, it's human ingenuity and a little bit of luck sparkled in with a little bit of luck. Yeah.
It's a little bit of inspiration, a little bit of freedom to do what you have, a bit of latitude, but also it works well when you know when to quit.
Yes. Very
early on. Things have a tendency to linger. If you can decide fairly quickly, no, this isn't going to work. Because it was all done over a short spell of time.
I think you had mentioned that it was 6 to 9 months, dependent for some of these projects. And then what happened next? What did you do next from moving on to all of these projects?
What happened next was the merger with SmithKline Beecham, who had a very substantial orphan receptor program with upwards of 100 people. But the merger introduced a new way of almost industrializing the process. So we had one division who would do screening, another divisions would do the disease studies, disease orientation, another division would do the cloning. And so molecular pharmacology, almost as a discipline, disappeared because you did pharmacology or you did molecular. And the group that had done that work were just basically cast to the four winds. I mean, half left, half found their niche. My particular niche turned out to be bioinformatics because that's not what I was trained in, but I could see that you could get new stuff out of informatics. I always liked discovering things, and me and a computer, I didn't need you can do that almost in your spare time. And that I went into informatics, and at the time, GSK, as it was then, initiated a massive genetics program. years before anybody else did, with Alan Roses, the champion of association genetics, as its head, who was probably doing him a disservice to say he was evangelical, but he was, and he swept us all along with that idea. Now, we were years ahead of our time. Unfortunately, we were also thousands of patients short in the analysis.
Wow.
We could rediscover ApoE in Alzheimer's, for example, ApoE4 and the subtype. Of all the other diseases we looked at, 1,000 cases, 1,000 controls wasn't even close. 1 million cases, 1 million controls might not have discovered some things. So the flood of new targets and wonderfully genetically validated GPCRs that I was hoping for never quite turned up. But all GPCRs I knew of, and the ion channels, and the transporters, and the ligands, and everything I could think of went into those screens. And some things have held up, but genetics I'm always expecting it'll be really big next year. And it never quite gets there. It could be there for right drug, right patient.
Yeah,
it could be. But the problem there is it's the old story of you identify exactly who your drug will work in. So your clinical trial is wonderfully efficient and very cheap to run, but then you can only sell your drug for those small number of people. Yeah. Or you identify a wider range, but your trial costs millions, but you can get millions back and cover your costs. Genetics is potentially one of those tools that can make the difference.
It's a tough one. That's why sometimes, patients who are low in numbers, who have a specific genetic disorder, it's very difficult to big companies would go after those disorders and try and treat those because it's not wide enough. It goes into the category of rare diseases, and then there's
sometimes the rare diseases like Bruton's kinase Gleevec story, where you can get a kinase inhibitor for one particular misshapen kinase that's oncogenic, and then all of a sudden you can extend that into other disease, the other thoughts, but into the other disease areas. But most of the time, that hasn't really quite worked.
Yeah, it is difficult. I feel like repurposing kind of compounds or repurposing discoveries from one field to another would work really nicely when it comes to a GPCR, or at least a family of GPCRs. Maybe a compound works in one context, but it has X number of side effects, but you could try and manipulate it in order to target another GPCR. But still, it's a tough thing to do. And then did you, once you went more computational after all the GPCR, molecular pharmacology GPCR projects, did you ever wish to come back and do more pharmacology, or how did that go?
How did it work?
Yeah.
Yes, I quite frequently wished I was back, but back didn't really quite exist. Things move on quite rapidly. We got very quickly, even while I was at GSK, to the idea that quite often they would rather have a protein structure than they would have a screen. Things move on. People would, certainly for microbiology, they'd much rather have the protein structure of a bacterial target than they would a screen run on that bacterial protein, because then they can actually design small molecules to fit.
Yeah.
And we've got routine GPCR structures now.
We do.
Which we didn't quite imagine. Things are not done efficiently. They're still done the same way, but there are many clever ways. The way that we managed to cleverly do things in a short window, people are cleverly doing that now on the next set of technologies and the next one after that.
Agreed.
It's still hard to get from lead to tool to drug, and target validation remains, despite the genetics and the power of it now, and the sheer number of people you can process, still not quite there.
Yeah, and maybe it's gotten easier to find that lead, but then moving to the next step is still as difficult as it was before.
It is. A proper target validation is still extremely difficult. I mean, you asked me on your little crib sheet, what would you do next? Which is the classic. It's very easy. I've got the tremendous advantage of being hindsight, but looking forward, you'd think structural biology would go back. I still think synthesis of information. I mean, so much as the literature is just so much bigger than it was.
Yep.
Then it's just massive. So, for example, when I was working at Glaxo, we had chemoinformatics, bioinformatics, and library services were three completely separate things, but they were all processing information, but they didn't speak to each other, which is quite ironic, really, until bioinformatics got put with chemoinformatics. All of a sudden, we realised we had loads in common. And then commercial, we found that we identifying targets from patents. People with patents, they write them deliberately obscurely, so you can't guess what the target is. So they don't. It's they're covered. So that's. I would still champion information, which is what I did in the latter stages of my career. And most companies have the same information, and there aren't that many secrets.
Yeah, I think
people think there are big industrial secrets, but on the whole, I found most people are working on generally the same ideas.
Yeah, and we just need to figure out what are the tools that would allow to kind of synthesize that information and give you the rundown and how you can go from point A to point B, from that lead to that validation. If there was any, what other tools and initiatives do you think we need to reach? What are the milestones we need to reach to kind of speed up drug discovery, especially when it comes to GPCRs?
I think, we've already mentioned structures, and the idea that once you have a structure, you can see possible ways. I mean, it's easier said than done. Even with the structure of the ramp CRLR, what makes the CGRP receptor, it's pretty hard to spot how you could get what residues you're going to block to stop that interaction, because one's allosterically affecting the other, and most of your structures are just a snapshot of the receptor flip-flopping in one form or other, and you need to catch it in just the form you need. What form do you need? Is that form a biased conformation that's going to lead you to, I've seen from your work, the idea of agonist trafficking. Can you send it down one pathway or another? There's a real dearth of drugs that have come from that, but it's there. A lot of academic labs work on it, but turning that from oh yes, we need this pathway, not that pathway, and we're going to get a drug out of it. That's been essentially impossible, or not worked well, not well enough to have the breakthrough that we'd all hope you'd get. So I think antibodies could, the whole Chris Tate idea of freezing the receptor in one format, and then you can raise an antibody to it. That antibodies could be, to GPCRs, another big step forward, just like they have been in general therapeutics. The biopharm industry has really, captured a large chunk of the best-selling drugs now.
But wouldn't you still need to understand which conformation, which interaction between the receptor and which G protein, or any intracellular effectors you would need to capture, so that antibody recognizes that disease state receptor?
Yes. I mean, you still need to know that. In my early academic days, I worked in a lab that was concerned with Graves' disease, which is multiple autoimmune disease targeted at the TSH receptor, GPCR. Didn't know what the GPCR was, what it looked like. Some antibodies stimulate the receptor, so people get hyperthyroid. They're driven. Others block it, so they're hypothyroid.
Interesting.
So, in theory, you could actually identify one form or the other, and try and nudge the receptor away from the autoimmune things. But the antibodies, the autoimmune antibodies, the ones we generate to ourselves, are such high affinity. You'd have to have a wonderful antibody to nudge it away from your own immune system. That's just one I could think of while you put me on the spot. But
I didn't mean to.
You can raise antibodies to GPCRs increasingly. I remember being asked, would ramps improve the chance of drug discovery? And to my embarrassment, I said, no, I couldn't really see how it would do it. And in fact, it completely revolutionized it. So don't necessarily say what I say.
Going back to that patent, definitely you should have put that into a patent.
There's more to drug discovery than filing a patent.
I'm sure there is, and there definitely is. It's interesting that so you went from an academic to then working in industry, and even in industry as a molecular pharmacologist, and then you kind of shifted to a bioinformatics and computational. But I want to take you back before. To a younger age as a teenager. Were you always interested in science, or how did you at all get into a scientific path?
I think like a large chunk of my generation, I went through a few years of wanting to be Jacques Cousteau. Then I went through a small spell with Lego and being very keen on being an architect or whatever. And I thought of doing medicine, as a lot of people think of doing medicine, until they actually understand what it involves and what the day-to-day business of writing up case notes are on exactly the same disease, month in, month out. And so I went in. I did physiology as a shortcut into medicine. I went to a very poor school, and they advised me that it was really hard to get into medicine, so I should really apply for just a couple of medical schools and then hedge my bets, with zoology. And of course, you got rejections for not being committed by return of post. Yeah. So I thought, well, this is terrible. My dad's not a doctor. I'm never going to get into medicine. I did physiology, which I loved. And that was the shortcut into medicine. If you could cut it in Newcastle University as a physiologist, they'd let you into medicine. But I didn't want to go into. Once I did my research project on TSH, pituitary hormone tumors, Met-Reg Hall, the other people in there, Maurice Scanlon, Jack Peters, and that was it. I was hooked straight into research. Never really looked back. And I had a job that was a hobby, which is just what you would want.
That you touch on an interesting point, having a job that's actually a hobby, because then you're just interested in doing it more and more, but you never feel like it doesn't feel like work.
No, it never felt like work. And even on things that weren't, I am a natural enthusiast, so I can find most things interesting if pushed. But GPCRs were the most interesting, because if you grasp how they work and the spread of them, you have an understanding of a lot of disease processes without really trying that hard. I'd find myself piping up in all sorts of meetings, just because I knew a little bit about one control system, but it was a really important control system. So yes, do a job you love. You're much more likely to be good at it.
And my next question takes me to what would be your advice to junior scientists who want to either contribute to the GPCR field, but also succeed as scientists, whether it's in academia or in industry? One would be do what you love.
Do what you love. If you can find a really good supervisor stroke mentor, somebody who can guide you through that. Nowadays, it's as much about being connected. We were joking earlier how many people we actually know in common, even though we've never met.
Yep.
I know half the people on your list. On now, not always well, but they're very nice people and really enthusiastic about it. And if you find a supervisor who can connect you with those other people, there is a community out there. Quite often, it's very difficult because what doesn't work isn't published. These background stuff, you can, if you're working in isolation, you can spend a long time working on something which has got not any chance of working, and somebody else knows that.
Yep.
And you're not going to find out until it's too late.
Agreed. You've touched on two very interesting points. One is not working in isolation, talking to people. That's very important. And when I was a graduate student, a friend of mine, we were joking about creating the Journal of Negative Results. We always thought it would be such a good thing to have that journal in order to make sure that any new student back at the time would not be steered into a project where it is already known that it just doesn't work, at least not the way they were thinking of doing it.
Yes. No, it is important. I mean, it's very difficult sometimes to make an impact in your own lab. It may sound a funny thing, but when we were forced to try and clone this second angiotensin receptor that never existed, the reason we were particularly driven was because another drug company said they'd found it. And so our executives thought, well, they've got it. They're a really good company. How come we're missing it? And they wouldn't believe us that when we're missing it because it's not there. It develops a self-confidence. And if I'd actually gone out and spoken to more people, I might have had lots of people who had told me it's not there. I wouldn't have had to be working so long on the mass oncogene, the world's worst angiotensin receptor, which might actually, there's still something in that though. I still cannot explain that piece of information. That will probably be the fourth bit. It's an old quote. One of the most interesting things about science is when somebody says, that's funny. You know, when they see something and they can't explain it. And there is a time to let go of those things because you can't explain everything. But quite often the discoveries come from those sort of, that's funny moments where you've seen something you didn't expect and you keep seeing it.
Yeah, this is...
I can't explain the angiotensin mass oncogene story to this day.
That's when you have to kind of trust your gut and make the decision of diving in deeper or moving on.
Or moving on,
to something
yes.
else.
More people help in that sort of, the more you share it around, you're very, very unlikely to give away the discovery of the century. You're far more likely to find out that it's going to save you because somebody has done it before, has seen it, has heard it, and those people are the people who are connected.
Agreed. And especially after the year that we've had, you mentioned, talking to people in the field and getting introductions. I think everyone has gotten used to, working from home or being in front of a computer. And I found that it was so much easier. At least maybe it's also a question of confidence, just sending an email to someone you respect, you like their work, just send them an email. Worst case that happens, they don't get to you or they don't respond, but it is very unlikely, I feel like, in the GPCR field that people won't at least give you 15 minutes of their time to just chat.
No, it is an important thing for the senior managers, whether it's academia or industry, that they're completely open to that sort of contact. Because somebody they may have the discovery of the century, or they may be embarking on a complete wild goose chase. But people need to talk about it. It's when you're sat in your room on your own, which we've all been doing, it's not quite so good.
We're good. Yes, I have one last question before I let you go. So, as I mentioned, you work in academia and industry, and then you also worked as a consultant. What did that look like? What would you, before you answer that question, what did that look like? And if a junior scientist or a scientist would want to move into consulting, what would be your advice for them?
I'm afraid to say, one of the best ways to get into consulting is to actually get a senior position in industry. Because then people assume that you've been there and you've done it before. Even though industry moves at such a speed, they're probably wrong. But you've had to go through so many filters to get that senior position. They think you're decisive and you know exactly what's what. And if you're asked to do consultancy, people do expect you to be decisive. And in my experience, they quite frequently have made up their mind beforehand. I've done several sort of site visits to places, and they said, "Oh, produce your report in a week." And I'd got home that evening, and they said, "Well, what do you think?" And I'd say, and they'd say, "Yeah, that's what we think too. Don't bother with the report."
Okay.
And so that's the sort of thing. There's a different sort of consultancy, which is the expert consultancy. The sort of how many matched case controls do you need to do a genetic study, and what sort of statistics do you need to work it out, because the numbers you're going to generate from that genetic study are so absolutely vast, you can't use your standard student's t-test. That's a different thing. I never had that. I'm always too much of a generalist. Apart from the CGRP story, for which I was a legal consultant.
Interesting.
Which was that's just luck. You discover something that turns out into a drug, so people will come back 20 years later and ask you to talk about the good old days. And you can hardly believe your luck. So I can't recommend that as a career path.
Definitely not, but I think it's still something that students, or at least, hire more senior postdocs or even PIs tend to forget that you're an expert at what you're doing. And
yes.
You have to trust that expertise. And sometimes as a postdoc, you're so deep into that topic particularly, is that you know more than basically a lot of people in the field. So I think trusting that experience and trusting what you know about that specific topic can also be a good way to try and think and become an expert consultant on that.
There are positions for that. I mean, I made the joke of genetics as an expertise, but our lab cynic who didn't believe the ramps were true until we proved it to him, it's called Steve Reese, who has a very senior position with AstraZeneca now. And we couldn't have done a lot of our work without him, as he devised all the vectors. And he's now high throughput screening, robotics. His career as a consultant would be stellar if he left AstraZeneca, and it's pretty good as it is. So that's there are positions. You would not have guessed, though, starting off, that you could make a career out of screening and robotics, and now it's routine.
It is. And
I mean, you mentioned it yourself. The screens, the systems, the high content screening. That's what he's an expert in, and it was invisible 10 years ago, but he was there at the start.
Which also means that whenever you like to do something and you're really good at it, just keep at it, because
he really loved it. He really loved it, and which is a good job, because we had a balance of people who love different things. That's very hard to put a number on that, though.
It is.
What is the right balance of people to make a team?
That is I think we would have to record another episode about how to put together a good team and how lucky you have to be. I think there is a big component of luck into bringing together the right expertise, but also the connection that has to happen between between the humans in that team.
Yes, indeed.
Super. Thank you so much, Steve, for your time. I really appreciate it.
Thank you very much. I've thoroughly enjoyed myself. I hope the listeners enjoy themselves half as much, which I suspect, but not. But anyway,
I think they will. Thank you so much.
Okay. Bye-bye. Thank you very much.
Bye-bye. Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, Alexa Duran, and Ines Pinero. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.